Lipids, most people just think of them as fats, but in reality, they are some of the most crucial molecules in the human body. They store energy, build cell membranes, and even transport essential nutrients in the blood. But what makes triglycerides different from phospholipids? Or how lipoproteins determine heart health? In this video, we're diving deep into the world of complex lipids, glycerol esters, phospholipids, sphingolipids, terpenes, and lipoproteins. Whether you're preparing for the ASCP exam, studying biochemistry, or a medical professional looking to refresh your knowledge, this guide is for you. We'll break down the structure, function, and clinical significance of these lipids, how they are digested, transported, and even how they contribute to diseases like atherosclerosis. By the end of this video, you'll have a clear understanding of why lipids are far more than just fat, they're biochemical powerhouses. So, get ready as we unravel the science behind the lipids that keep your body running. Let's start with glycerol esters, also known as acylglycerols. These are complex lipids in which fatty acids are covalently linked to a glycerol backbone, a 3-carbon molecule with alcohol, OH, groups. This simple yet versatile structure plays a fundamental role in lipid metabolism. Classification of acylglycerols. Glycerol esters are classified based on the number of fatty acids attached. Monoacylglycerols, monoglycerides have a single fatty acid attached to one of the 3 carbon atoms of glycerol. Diacylglycerols, diglycerides contain 2 fatty acids. Triacylglycerols, triglycerides have 3 fatty acids linked to the glycerol backbone, making them the most common and important form of stored fat in the body. The specific carbon positions where fatty acids attach are denoted using a stereospecific numbering system, alpha, SN1, beta, SN2, and alpha prime, SN3. This is important because the position of fatty acids influences how they are digested and metabolized. The importance of triglycerides. Triglycerides make up about 95% of tissue fat and are the main component of stored energy in the body. They also form the core of lipoproteins, which transport lipids through the bloodstream. The composition of triglycerides varies by source. Plant-based triglycerides are rich in unsaturated fatty acids, like linoleic acid, C18-2, and tend to be liquid at room temperature. Animal-based triglycerides contain more saturated fatty acids, C14-0 to C18-0, making them solid at room temperature. Exceptions exist, such as coconut oil, which is plant-derived but rich in saturated fats, making it solid at room temperature. Understanding the role of glycerol esters is key to grasping lipid metabolism. Next, let's explore how triglycerides are digested and absorbed in the body. Triglyceride digestion occurs primarily in the duodenum and proximal ileum, where enzymes break them down into smaller components for absorption. Emulsification by bile acids. Before digestion begins, bile acids from the liver break large fat droplets into smaller micelles, increasing their surface area for enzyme action. Enzymatic hydrolysis by lipases. Pancreatic lipase is the key enzyme responsible for breaking down triglycerides. It hydrolyzes triglycerides into monoglycerides, free fatty acids, and glycerol. This process requires cholipase, a protein that helps pancreatic lipase function efficiently. Absorption and reassembly. Once broken down, monoglycerides and fatty acids diffuse into intestinal epithelial cells. Inside these cells, they are reassembled into triglycerides and packaged with cholesterol and ApoB48 to form chylomicrons. These chylomicrons enter the lymphatic system before being released into the bloodstream for transport. Triglycerides are the body's primary energy storage molecules, making their digestion and transport essential for metabolism. Next, we'll explore another vital lipid class, phospholipids and their roles in cellular function. Phospholipids are more than just fats, they're essential for cell membranes, signaling pathways, and lipid transport. Unlike triglycerides, which store energy, phospholipids have a unique structure that makes them both hydrophobic and hydrophilic, allowing them to form the foundation of biological membranes. Structure of phospholipids. Phospholipids consist of a glycerol backbone, just like triglycerides. 2 fatty acid chains, typically with a saturated fatty acid at position C1. An unsaturated fatty acid at position C2, which increases membrane fluidity. A phosphate group at C3, which is linked to a variable alcohol-derived group. Types of phospholipids. Phospholipids are named based on the group attached to the phosphate. Phosphatidylcholine, lecithin, the most abundant phospholipid, crucial for cell membranes and lipoprotein transport. Phosphatidylethanolamine, found in brain and nervous tissue, important for membrane curvature. Phosphatidylserine, involved in cell signaling and apoptosis, programmed cell death. Phosphatidylinositol, a precursor for second messengers in signal transduction. Specialized phospholipids. Some phospholipids have unique roles. Cardiolipins, found in the inner mitochondrial membrane, essential for ATP production. Lysophospholipids, phospholipids missing one fatty acid, important in inflammation and lipid metabolism. Functions of phospholipids. Membrane structure. Phospholipids form bilayers, creating a selectively permeable barrier for cells. Lipid transport. They stabilize lipoproteins like HDL and LDL, which transport cholesterol in the blood. Cell signaling. Phosphatidylinositol derivatives act as second messengers in hormone and neurotransmitter pathways. Phospholipids are fundamental to membrane integrity, communication, and lipid transport. But beyond phospholipids, another class of lipids, sphingolipids, play a critical role in nerve function and cellular interactions. Let's explore their structure and significance next. Sphingolipids and their biological roles. Unlike glycerol-based lipids, sphingolipids are derived from sphingosine, a unique 18-carbon amino alcohol. These lipids are essential components of cell membranes, particularly in the nervous system, where they play key roles in signaling, cellular interactions, and disease mechanisms. Structure of sphingolipids. Sphingolipids consist of sphingosine backbone. Instead of glycerol, sphingolipids are built on this long-chain amino alcohol. Fatty acid chain, attached to the sphingosine backbone via an amide bond, forming ceramide, the simplest sphingolipid. Head group, this determines the type of sphingolipid. Types of sphingolipids. Sphingomyelin, the only sphingolipid that contains phosphocholine, making it both a sphingolipid and a phospholipid. It is a major component of myelin, the protective sheath around nerve fibers. Cerebrosides, contain a single sugar, galactose or glucose, found in neuronal cell membranes. Gangliosides, complex sphingolipids containing multiple sugars and sialic acid, highly concentrated in the gray matter of the brain. Sulfatides, formed when a sulfate group attaches to galactose, playing a role in nerve conduction. Biological roles of sphingolipids. Neural function, essential in myelin sheaths, ensuring efficient nerve signal transmission. Cell recognition and signaling, glycosphingolipids on cell surfaces regulate immune responses, growth, and differentiation. Disease markers, abnormal ganglioside accumulation is linked to neurological disorders like Tay-Sachs disease. Clinical significance. Blood group antigens, glycosphingolipids contribute to ABO blood typing. Tumor markers, some glycolipids act as tumor antigens, making them targets for cancer research. Sphingolipids are critical for nerve function, cell signaling, and immune responses. But beyond these, another class of lipids, terpenes, play fundamental roles in vitamins and biochemical processes. Let's explore them next. Terpenes might sound unfamiliar, but they are everywhere in biology, from vitamins to essential oils and even in our cell membranes. These lipids are built from isoprene units, which are simple 5-carbon building blocks that can polymerize to form larger structures. Structure of terpenes. Terpenes are classified based on the number of isoprene units they contain. Monoterpenes, C10, found in essential oils, such as limonene in citrus peels. Sesquiterpenes, C15, found in plant hormones and defense compounds. Diterpenes, C20, include vitamin A, retinol, which is essential for vision and immune function. Triterpenes, C30, form steroids and cholesterol, key molecules in metabolism. Polyterpenes, large chains of isoprene, like rubber, used in biological elasticity. Functions of terpenes in the body. Vitamin synthesis. Terpenes are the backbone of vitamins A, E, and K, crucial for vision, antioxidant defense, and blood clotting. Steroid formation. Cholesterol, the precursor for steroid hormones, is derived from triterpenes. Protein glycosylation. Dolichols, terpenoid lipids, help in attaching sugars to proteins, a process essential for cell signaling and immune responses. From vision to metabolism, terpenes are essential biochemical building blocks. But how are lipids actually transported in the body? That's where lipoproteins come in. Let's break them down in the next video. If you found this video helpful, hit the like button, subscribe, and share it with your peers. Thanks for watching and see you in the next video.